Right, so this presentation is on transport in plants, which is unit 3.3. So we start straight away with the xylem. So the xylem carries water and dissolved mineral ions. Okay, it's got fibres to support the plant. It contains next to it living parenchyma cells, which act as a packing tissue, which helps separate and support the vessels. So these parenchyma cells are really essential for the xylem to function. The most important thing about the xylem is the lignin. So xylem cells, at one point they were living cells. Then lignin impregnates the cell walls. It causes all the cytoplasm to dry up, the cell contents to die. It forms end walls, and it is waterproof. So the cells in the xylem, they're all dead. Lignin prevents the column from collapsing, and then it forms annular or reticulate rings, which help give the plant flexibility. In some places there is incomplete lignification. This forms bordered pits. Okay, so bordered pits are between the xylem vessels. So you might have two xylem vessels here next to each other, and the bordered pits are like holes in between the two, from which I'll get a nice blue colour for this. Water can move between the two xylems. Okay, so these are two xylem vessels here. So water can move in between those two vessels. The bordered pits are really essential for the water columns, which I'll speak about later, to be maintained. All right. Then we've got the phloem, which transports assimilates and such as sucrose dissolved in water, which forms sap. I'll write sap down because that's an essential word you might see a lot in the exam. There's two main parts of the phloem. You've got the sieve tube elements, which have no nucleus, very little cytoplasm. The sap moves through these sieve tube elements by mass flow. At the end of each sieve tube element, instead of xylem, so xylem we've got, if we go back, xylem are long narrow tubes. They've got no end walls, so there's nothing separating them. That doesn't exist. But in the phloem, we've got each single sieve tube element has an end wall, like so several end walls. And these end walls are perforated, so that means they've got holes in it. So if we have a cross section of it, there'll be lots of little holes in this end wall. Okay, so that allows the sap to move through, but by having the end walls, it can control the movement of the sap down the plant. Otherwise, it would just rush down to the bottom. You wouldn't be able to load assimilates into it, etc., etc. Sieve tube elements they've got very thin walls, and next to them they've got these companion cells. So companion cells are in between the sieve tubes. They're small cells with a large nucleus and dense cytoplasm. So pretty much opposite to the sieve tube elements, which are pretty much hollow. They've got numerous mitochondria to produce ATP for active processes. So the phloem, as I said, transports assimilates, and to get these assimilates into the phloem from the source to the sink, we need to actively load the assimilates in. So active processes, as we know, involve the use of ATP. Let's write ATP. Okay, which leads assimilates in the sieve tube. Right, so companion cells, sieve tube elements, they work together, and they're just like xylem as well. We have parenchyma, so the packing tissue to help give support to the phloem itself. Okay, so they don't collapse. Right, then we're going to look at vascular bundles. So vascular bundles, here we're looking at dicotyledonous plants, their arrangement is for strength and support, and they also contain meristem cells. So when we're talking about vascular bundles, we're going to look at the root and the stem and see the arrangement of the xylem and the phloem, because they are quite different and it is for different reasons. So let's first of all look at the root. So here is a root. I've already highlighted the medulla, which is the space inside the cell, and the endodermis, which is the wall around it. So the endodermis is a special sheath of cells which contains meristem cells called the pericycle. So meristem cells, as we know, are like stem cells. They can differentiate into any type of cells. So the endodermis therefore supplies new phloem and new xylem cells. Then we've got the xylem, which is in blue. Xylem. That's in the center of the arrangement. And then we've got outside of that, we've got the phloem, which are these red dots. Phloem. Helps if I zoom in to right and it looks a bit neater. I'll remember to do that next time. So that's really basic. That is in roots. This arrangement, they're all tightly compact. That's a really poor circle. I don't want to do it in red. They're really tightly compact together. As you can see, they're all tucked together. It's because they need to be packed together because roots have very high pressure because they're trying to get lots of waters flowing through them. They have to withstand the pulling forces which roots are exposed to. So we've got a tight pack. Let's get rid of those two circles. Now we're going to look at the stem. The stem is a bit more complicated. As I said before, in the roots we just had the medulla. We didn't have this thing called the cortex. So we've got the medulla in the middle. So medulla, middle of all these things here, these little bundles that I'm about to explain to you. And outside of that we've got the cortex. And there's a reason for this. While it looks like this part of the same thing, medulla and cortex can have different water potentials, different concentrations of assimilates, etc., etc. And it is really important for these two things, for movement. Then here we've got the sclerenchyma, which have lignified walls. Okay, so let's zoom into this one. It's not very neat, but this green blob here, this green is the sclerenchyma. So they've got lignified walls to provide strength to the stems. We've also got the phloem in red again. So let's write that down. Phloem. As you can see, they're in the middle between the sclerenchyma in green and the xylem, which is in blue. So let's write xylem. Right. So that looks simple, but there is a bit more to it. So if we zoom in on this one, where we've got the sclerenchyma, the phloem, and the xylem. This bit in between the two, let's get a different colour for that. Let's go purple. This line in between the two is called the cambium layer. Okay, cambium layer. And the cambium layer is in between the xylem and the phloem. It has meristem cells in it, which provide new xylem and phloem. So this here has meristem cells to provide new xylem and phloem. The bundles can separate in young stems, but become a continuous ring in older stems. They can create a complete ring of vascular tissue underneath the bark, providing strength and flexibility to withstand the bending forces in the tree. If we were to cut, I'll take that line out, if we had a tree here, and we cut it across there, we're looking at that cross section, which is this, alright? And that is the vascular bundle in the stem. Right, now we've nailed what the xylem and the phloem are, we can look at the movement of water throughout the plants. Right, so there's three ways you can see the apoplast pathway, which is, one way to remember it is around. So this is when the water moves through cell wall spaces and between the cells via mass flow. Okay, there's one thing we're not drawn on here. Oops, that was way too big. Let's get that down. One thing we're not drawn here is little breaks in between the cell walls. And these are called plasmodesmata, so they're gaps in between two cells, allowing movement of water in between cells. Alright, so the apoplast pathway is around the water, just moves through the cell wall spaces and in between cells. So water can strew a pathway here, goes down here, goes through the plasmodesmata. It never actually enters. Never enters within the cell membrane. Right. Then we've got the symplast pathway. So the symplast pathway, one way to remember that is simple. Let's do this in a different colour. Simple. Right. To you that might seem ridiculous, but it helps me remember it. Simple, basically it goes through everything but the vacuoles. Through plasmodesmata, absolutely anything and everything it will go through. So it goes through the cell cytoplasm and plasmodesmata between cells. Okay, and then our final one is the vacuola, which clues in the name, it goes through the vacuoles. So basically, vacuoles, I've drawn really small ones here. It goes through, basically straight through the middle. That's the vacuolar pathway. It goes through plasmodesmata, basically anything apart from the double membrane organelles. So they're the three water pathways. One thing to look at now is water potential. So water potential is the measure of tendency of water molecules to move from one place to another. So they always move down the concentration gradient. Pure water has a water potential of 0. Water potential equals 0 in pure water. Here we've demonstrated how it moves from a high water potential to a low water potential. That way, okay? It never, draw this in red, never goes that way. Alright, let's get rid of those arrows to make it look a bit neater now. Right, turgidity, which is really poor handwriting, and plasmolysis, okay? Turgidity is created when you've got a high water potential outside the cell. So water moves in to the cell, okay? This causes the cell to become full of water. Water exerts pressure on the cell wall until it reaches a pressure potential. So this pressure potential, if we get rid of these lines, is the cell membrane pushing back against the cell wall. Eventually, the pressure potential going out, pushing outside, will equal the water potential pushing this way. So they equal each other, okay? That's a really poor arrow. Then we've got another thing, plasmolysis. So when the water potential outside the cell is less than inside the cell. So water obviously wants to get to the low water potential here, so it moves out. Okay, this causes plasmolysis. Okay, so in isotonic solutions where we've got a low water potential, the cell wall will lose water via osmosis down the concentration gradient, as the water loss increases, the vacuole and cytoplasm shrink, as shown here. The cytoplasm stops pushing on the cell wall. The plasma membrane loses contact with the wall, which is plasmolysis. So if all cells do this, the tissue becomes flaccid. Right, next we've got transpiration, which is the loss of water from the upper parts of the plant. Most is lost through stomata, which are open for gaseous exchange. However, the waxy cuticle reduces water loss as it prevents too much water evaporating from the upper surface. So we're looking at the lower surface of the plant where we see the stomata. Most water is lost during the day when photosynthesizing, as the stomata are open during the day. So as we know, light causes the stomata to open. So how does transpiration work? So water enters the leaf from the xylem. It enters the spongy mesophyll layer, which you should recognise. It's a few cells, lots of air spaces in between them, and the air and water vapour can move in between them. So the water evaporates from these cell walls, these cells of the spongy mesophyll layer, and then it moves out via stomata. So if I draw stomata, obviously we know they're guarded by guard cells, which can open and close them. Stomata are gaps, and the water vapour can leave the stomata. But this only happens, transpiration only occurs if we've got a lower water potential outside of the cell. Okay. The importance of transpiration, like why would we want to lose water? So mostly it is for the transpiration stream. So if water did not move out of the cells within the leaf, we would not be able to pull water up the leaf. We're going to speak about that in much more detail later. So it also is important to maintain turgidity and cool the plant. So transpiration stream, let's write that down. Transpiration stream. That's the importance of it. To draw water up the xylem. Okay, let's get a different colour because now we've got a lot of clashing. Green will do fine. Right, so we've seen the importance, but what factors affect transpiration? And first of all, we'll go to light intensity, okay? So the higher the light intensity, the more stomata will be open. So hence, during the day, we have a higher rate of transpiration. Okay, so the more light, the stomata open, and then more water can be lost. Okay. Next, we've got temperature. So temperature, as we know, increases the kinetic energy of molecules. So we've got higher kinetic energy, so hence higher rate of evaporation. And temperature causes a decrease in water potential in the surrounding air. So water moves out of the leaf into the air. So high temperature, high light, they both increase water potential. Then we'll look at wind. We'll leave humidity to last. No, actually we will look at humidity because it's essential for wind to be understood. So humidity, if we've got a high water potential surrounding the leaf, which is humid air, lots of water surrounding the leaf, the water will not leave the leaf. So high humidity, let's write this in this little gap, high humidity causes a low rate of transpiration. I'm not going to write it all out, okay? So we've got a high humidity, we've got a a low rate of transpiration because the water does not want to move out to a higher water potential. Then this is where wind comes in. So it could be humid, but if it's windy, then the humid air surrounding the leaf will be moved, and it reduces that gradient, so water will then move out because we've not got humid air surrounding the leaf, we've not got a high water potential surrounding the leaf, so the water moves via diffusion out of the leaf. Then a final one is water availability. In low water availability, the stomata will close, which will lower the rate of transpiration. Okay, it's pretty simple. If you've got no water, you've got no light, your stomata will close, and no water will be lost via transpiration. Some may be lost if you've not got a thick waxy cuticle through the upper surface of the leaf. Now let's look at photometers. So they measure the rate of transpiration. Measure the rate of transpiration. Rate. Trans. Okay. It's a really simple setup, but first of all you must have no air bubbles in the setup. So we do this by cutting the stem underwater and setting up the apparatus underwater. We measure the movement of the meniscus from the start of the measuring device here, which would be a ruler, and what we're doing to see is the amount of water uptake by the plant. So the more water uptake by the plant, we relatively think that the plant will be transpiring more. It's a fact. The more the plant transpires, the more water it will uptake. So while this is not actually telling us the rate of transpiration, it's telling us water uptake. That's what you've got to remember. Water uptake. Not actually the transpiration rate, but we're trying to work that out. We want to work this out, but we have to measure the water uptake. Okay, so we only want this meniscus here. We don't want any air bubbles. We need the plant to have dry leaves, because if it's got wet leaves, obviously no high water potential, no water will move out. So we need to cut the plant underwater, apparatus underwater, for it to work. Okay, you should look at that in a bit more detail though.